video on the glymphatic system, I'm going to focus on the blood-brain interfaces. In the previous video, I talked about kind of an overview and the difference between the body with a one-way open-ended lymphatic system and a closed-loop glymphatic system paralleling the entire vasculature of the brain. And the glymphatic system is a concept first described by the group von Meijk and Nedergaard, and they described how CSF enters the brain parenchyma, and they demonstrated that by using different sized tracers that were injected into either the cistern or the ventricular system, and they showed how these solutes entered the brain and were also cleared by the brain again. And one of these solutes was amyloid, which is important in Alzheimer's dementia. And there was a lot of focus on the waste clearing of the glymphatic system in the first articles. And the glymphatic system was called the garbage truck of the brain. And not all amyloid is cleared by the glymphatic system. It's also partly taken up by the blood-brain barrier into the blood plasma. The glymphatic system can also be demonstrated with MRI. If you inject gadolinium intrathecally, it enters the brain parenchyma and gives a change in the T1 signal of the brain parenchyma. And this is a study in patients with normal pressure hydrocephalus. You can see the enlarged ventricular system. And in these patients with normal pressure hydrocephalus, you can also make maps of the change in T1 signal over time and see how the gadolinium goes through the brain parenchyma. And there's some CSF entering the brain periventricular, but that is because these patients have normal pressure hydrocephalus. Normally you don't see that, but what you do see is that there's entering of the gadolinium into the brain in a centripetal manner. And you can also see that it's mainly near the anterior and middle cerebral artery, and then it goes to the more central parts of the brain. So if you look at a microscopic level at the blood-brain barrier itself, you have the endothelium of the blood vessels, you have the CSF in the perivascular space, and then you have astrocyte and feet surrounding this entire complex. And the CSF in the perivascular spaces has a very constant composition. We already talked about CSF production in one of the previous videos, but if you look at how CSF is composed, it has a low protein content, a high iron content, a very stable potassium level, and the composition of CSF is pretty similar across species. And CSF is produced in the choroid plexus by very leaky capillaries, so all the large molecules enter the stroma, and then the epithelium cells determine which large molecules are going to enter the CSF. And if you look at these genes, they are very evolutionary conserved, so they're the same in zebrafish, mice, rat, and humans. And if something's evolutionary conserved, it means it's very important. And I also already mentioned that we need a very constant composition of CSF because the neurons need to be able to build action potentials across their membranes. So the extracellular space has to be very constant because otherwise you cannot build action potentials. You have a lot of interference. I also said on the website that in the rest of the body, capillary walls consist of one layer of endothelial cells, and in the brain you have, besides this one layer of endothelial cells, also the perivascular space and the astrocytes before you can enter the interstitium. And I mentioned when we talked about hepatic encephalopathy that the glucose is taken up by the astrocytes from the blood plasma. It is, but it is good to keep in mind that between the blood plasma and the astrocyte end feet, there's always, even if it's microscopic, some CSF, some perivascular space. And you can also see that on this electron microscopic image. So you have the lumen of the blood vessel here. This is the endothelium of the capillary of the brain with these tight junctions like tiles of the roof. You can see a pericyte here. Then the purple are the astrocytes, and between the astrocytes and the endothelium are two basal membranes and some CSF, and that's your perivascular space. So the small molecules can go from the blood plasm, such as oxygen and glucose can go from the blood plasm to the brain parenchyma. The large molecules are mainly derived from the CSF in the perivascular space, so they don't come directly from the blood vessel here. They have been excreted at the level of the choroid plexus. So the glymphatic system is also important to provide nutrients for the brain. And this glymphatic system, when it forms, first you have the angiogenesis, we talked about that before, and then the astrocyte end feet kind of zip and seal the entire vascular tree, and then it's matured and maintained during life, and that's your blood-brain barrier. The blood-brain barrier is not only important for waste and for providing nutrients for the brain, it also has a very important function in neuroinflammation and the immune system. So there's always some white blood cells in the perivascular spaces, and they are kind of patrolling the borders of your vascular tree. And if you, for example, have a cut in your toe, the white blood cells in your CSF go up to make sure that the infections do not enter the brain. So it doesn't matter where in your body you're injured, there's an immediate response of increase in patrolling white blood cells in your perivascular spaces. So I think that's kind of cool, and we're going to talk about the neuroinflammation later. But first, we're going to talk about enlarged perivascular spaces.